I was really asked to give a basic introduction because not everybody here is a cryo-EM expert, and so I was asked to do a little bit of an introduction to the methods. I'm also not a hardcore algorithms person, but I'm going to talk about what I think we need as in a practical sense. So I'll give a little introduction to what is this cryo-EM thing, where I think we need some work, and I'll show you a couple of case studies of places where we are really sorely in need of better algorithms. Most of what Clint and I do at the SAM in our research is to develop automation, so it has a little bit of a bent towards automation. But I thought the first slide should be, why do we even care? Why do we care about cryo-EM these days? And I think it's quite interesting. This is 2014, that's 2019. And these are the number of depositions into the PDB for integral membrane proteins. Integral membrane proteins being accounting for more than 50% of drug targets at the moment. So since 2014, this was cryo-EM, this little pale blue section. This year, cryo-EM overtook X-ray crystallography in depositing more PDBs for integral membrane proteins in X-ray crystallography. And that is an astonishingly rapid overtaking, I think. So it's quite clear that the method is very important. It's very important for drug discovery and drug development, and that's basically why we care. So what is cryo-EM? Lots of you know this. I'll just do a quick introduction for those who are less familiar. There's two different versions of it now, and this gets muddled up all the time. There's single particle cryo-EM, and then there's cryo-electron tomography. We care deeply about both of them, and both of them need a lot of algorithm development. This is even a little bit more complicated. It has a method called fib-sim milling and a cryo-light microscopy as well. So it gets to be quite complicated. But anyway, basically, I'll do a very dumbed down version, much more dumbed down than Joachim's fancy manifolds. But how we think about cryo-EM is basically you have single particles that have already been purified out of the cell. You put a little solution of this onto the grid. You reduce that to a thin film and slam it into a really good cryogen. You heard Fred asking Joachim, what's the temperature change during this process? We kind of believe it happens very, very rapidly, so it snaps from the liquid to the solid state, and we have all these little conformations trapped just as they were. That's probably not correct, but it's almost correct. So then we take this thin, trapped film of particles and we stick it into a transmission electron microscope. The transmission electron microscope, that means the electrons go through the sample, means the sample has to be very thin, otherwise all of our assumptions break down. And so the electrons go through, they produce a projection image of the sample, and it looks something like this. We have all these different views of the particles, and that's just because, well, we hope we do get lots of different views as they're tumbling around in solution and they got snapped into that solid phase. We get different projection views of the sample. It's also incredibly noisy. This is an enormous particle, so we can see it really well. Our very tiny particles, I'll show you later, you can barely see it all. But we can get around the noise by picking lots of self-similar particles, and this was what Joachim was talking about before. We can cut them out of our images, and now we have to rotate and translate them into the same positions so that we can average them together and get around our signal-to-noise problem. So that's basically what we're doing. But this translation alignment and rotational alignment has to be very good, and that's partly what Joachim was mentioning before. And of course, it's never quite good enough, but it gets better every year. If we can do that for one view, we can do it for all the other projection views. And as as everybody understands, if we have many projection views of a three-dimensional object, we can get the object back, essentially using tomographic methods. So that's in the simplest way you can think about it, single particle microscopy. This method called cryo-electron tomography, we're really interested in, because what happens if the molecules I want to see are inside a cell? They're not purified, they're not in a nice solution, they're inside a big fat cell, not such a big one in this case, a bacteria. Well, if it's small enough, we can use exactly the same method. We can just quickly plunge freeze it, and here's a little E. coli frozen. What we do then is we take that, and this is just the team of people at the Simons Electron Microscopy that are working very, very hard to get these methods established at the moment. We take that frozen cell now, put it back in the electron microscope. But now we can't take a single projection image because it's a very cluttered environment. We'll be looking through many, many, many different things, and we won't be able to sort it out. So instead, we use a method called tomography. cryo-electron tomography, and we simply take that sample and we tilt it, snap an image, tilt it, snap an image, tilt it, snap an image, just as you would in a CT scanner, except you'd be stationary and the machine would be moving. We move the sample and the machine stays stationary. So you get this tilt series, and again, okay, so that's a projection series. We know from a projection series we can reconstruct a three-dimensional volume, and that's what we get. One more complication: what happens if the cells are very thick? They will not vitrify just by slamming them into liquid ethane. They'll form ice crystals and be destroyed. In that case, we use a thing called a high-pressure freezer, which can take much bulkier samples and snap them into that vitrified state. But that's now I'm looking at this on a scanning electron microscope. Those are much too thick to get an electron beam through in a transmission electron microscope. So now you've got this thick vitrified sample and you have to thin it down. What we do is we put it inside this instrument called a focused ion beam scanning electron microscope. And what we do, and this is a nice little movie courtesy of Thermo Fisher, what we do with the focused ion beam is chop away a lot of the bulk sample, and we leave a little thin window that's left in the cell, thin enough now that we can get an electron beam through it in a transmission electron microscope. That looks very pretty. This is what they look like in practice. Actually, that's a really, really nice one. You can see these yeast cells embedded in this thin little window that's left after we chop away most of the cell, and then we do the whole thing all over again. We stick that inside the transmission electron microscope, and we take these tilted views, a tilted series of images. This is much more typical, lots of contamination. It's quite a tricky method. And then we take that and we turn it into a three-dimensional map again. And of course, what we want as molecular microscopists is we want the molecules out of that map. So you can go in and find them. That's actually what a nuclear pore looks like in this very noisy map. And those are some microtubules. And there's a ribosome in the cytosol, and there's a ribosome in the mitochondria. In theory, we can pull those out and do some averaging. This is very beautiful work, mostly from Wolfgang Baumeister's lab, where you can pull out these individual molecules from those very noisy maps and then look at different conformers of them and infer something about what they're doing in the cell. But man, that's jolly hard, and I think that's one of the challenges that we have, and one of the very interesting things that remains to be done. Just a quick brief history. We did this for many, many years. In 1985, there was the first Gordon conference on this, and people were talking about vitrification as this brand new method. And nothing happened much except in a few hundred people until about 2014, where there was the so-called resolution revolution. And it was culminated in the development of some new cameras, that did something special. And our association with the works, because Nico Grigoriev spent a year on sabbatical with us, so we got a whole bunch of work done around those new cameras. Just quickly, what were they? Prior to about 2013-14, we were using CCD cameras, just the same thing as in your cell phone or anywhere else. CCD, and we produce electrons, but CCD cameras require photons, and so we were using scintillators and fiber optics to turn those electrons into photons, and as a result, we had a lot of blurring of the individual electrons. These new direct detectors detect electrons directly. And so here was one of the very first things we did in about 2013. This is the same image on the old CCDs and the new DDDs. They look very similar as an image, but if you take it into Fourier space, you'll see that the signal goes much further out in Fourier space for the direct detectors. That's a direct result of not having that fiber optic scintillator mess of scattering anymore. But it was much better than that, in that the new cameras also read out fast enough that they gave us movies instead of individual frames. And it had been known or suspected for a very long time that while we were taking our images in the electron microscope, the whole thing was moving around. Maybe the whole grid was moving, and then the little particles that you're trying to see were moving independently of that. And because of these new direct detectors, we could now observe that. So the images we now collect are not a single open your shutter and shut it. We get this whole stream of images, and then we simply add those up to form our final image with enough dose that we can see anything. So we have this set of images. We can watch what happens while we're exposing our delicate biological sample to the beam. And there's a counter here going 1 through 6, 1 through 6. There's actually 60 frames, but I've added the first 10, the second 10, the third 10, because otherwise you wouldn't see anything. The signal is so low in these images, you wouldn't see. And you get the impression of a lot of movement here. We can put down some fiducials. Here's the stationary edge of the substrate holding our sheet of ice. And you can see these little particles are bouncing around like crazy. We can use cross-correlation from frame 1 to 2, 2 to 3, etc., to measure how much they're moving. And these are the vectors of what they're moving. And this scale bar here shows that many of them are moving by 25 angstroms. So it is small wonder that until this revolution came along, we were not getting atomic resolution structures out of images like these. I mean, if you want a 3 angstrom structure, you better not have movement of 10 or 20 angstroms through a structure. And the only reason we're getting anything out of EM was because we were averaging our way out of it, averaging millions and millions of particles, getting the ones that were stationary and throwing the rest away. So because of that particular movie camera and good camera, we went from very blurry images like this. This is what we were stuck with prior to that. And then we went to nice, sharp, crisp images like this, because if we can measure the movement between each frame, we can correct for the movement. We can just realign them and add them up that way instead of add them up in this blurry way. And this is one of the first things we did in the lab in 2015, I guess. And this was a sub-3 angstrom map done by cryo-electron microscopy. And you could see side change, you could see water molecules. This was incredibly exciting. Never thought we'd live to see the day. Now, of course, sub-2 angstroms is what you have to get and for our very good samples. And this is our favorite apoferritin. And at 1.9 angstroms or so, you can see every side chain and many of the water molecules. And it really, truly is atomic resolution. I won't be talking about sample prep. That's one of the kind of unsolved problems, and I think is why many, many samples do not get to sub-2 and are more stuck in the 3 to 4 angstrom range. And just a quick word, though. The resolution revolution, as it's so called, the direct detectors get a lot of credit for that, but there were lots of other things going on at the same time. The microscopes we use now are astonishingly stable and robust instruments. I mean, they're stable thermally, mechanically, electrically. We can run them for days and days on end. And they're super complicated machines, but they don't fail that often. Their uptime is very good. So that was a huge advance. And then computers was advanced enormously, of course. That first 3 angstrom structure we did in 2014 or wherever it was took us 10 CPU years. We begged and scraped every CPU at Scripps for 3 months to try and get it done in time for a meeting. I'm sure you can do it now in 20 minutes, and that's a direct result of the huge advances, especially in GPUs, which the gaming industry has driven. And so now we're doing everything incredibly fast. And then software was tremendously important. Automation software is important because some of these big structures that we do require 17,000 images, not 17,000 particles, 17,000 individual images snapped at the electron microscope. And then you pull particles out of there, 2.8 million particles in this case. So we have to automate that whole process. And my group did that many, many years ago. There's now many other packages to do that too. It's kind of a solved problem. I don't think anybody sits at a microscope and does anything manually anymore. It all gets done automatically. And then these software to correct for that motion, they have gotten better and better and better. Every year they get a little better, and we squeeze another half angstrom out of our structures. And I think there's still room to go and room to improve how to get that alignment on these very, very noisy images from image to image to correct for that. And then the really huge advance, I think, has been from packages like Relion and FreeAlign or System and CryoSpark, where you can take this noisy mess of tiny, tiny little particles that you can barely see, in this case was 2.8 million, and clean it up and find the 5% that actually contribute to your map and do that cleanup. But I'm sure there's a ton of work to be done here, and I'll mention that again in a minute. And also, of course, we can sort out many, many independent structures because of the software just from a mixed up series of individual conformers. And I'll mention that briefly. So basically, in the 30 years of cryo-EM that I've been associated with it, this was 1985 at that very first Gordon conference. This was the frozen hydrated ribosome. This was a 2D crystalline array of frozen hydrated ribosomes, and that was a 3D map that we got from it. We were super excited. Many years later, 30 years later, this is one of Joachim's maps, I think, a 2.5 angstrom map of ribosomes where you can see every water molecule. And I think none of us really quite believed we would get from there to there at the time. Maybe Richard did. And now, of course, because of these things where we can go into the cell and dig out the molecules, what we really hope sometime in the future is that what we're doing is cataloging at high resolution molecules out of the cell and finding out what they do and where they are. Okay, so our role has been a little bit to build automation here. So, our concept of this is we start with a sample, and maybe that's a cell these days, but at this moment, it's just molecules tumbling around in solution, and we need to end up in a map, with a map. So you have to prepare those grids, you have to make those thin films. I won't talk about this because I think this is an algorithms meeting, and there's not too many algorithms there, it's just machinery. Then we have to collect the data, and this, I think, is more or less of a solved problem. Maybe you could collect slightly better data, but the instruments are actually exquisitely good. But then this whole processing pipeline, where you have to do a whole bunch of things, that's really where we could use a lot of work. So that processing pipeline, at least in the Simons Electron Microscopy Center, looks like this. Here's a typical image, and we stick the sample in. We start imaging, say, 0 at time 0. After a couple of hours, we have a few hundred images, say, in 2 hours. That's about right. And while we collect the images, we already start processing. So we pick out those individual particles in those images. We correct for that motion that you see frame to frame. We have to correct that so they're nice and sharp. We do some other stuff, measuring some of the aberrations in the images that we need to correct for. This is also getting better and better. And then as soon as we have enough particles, we start making these 2D class averages, and then we turn those 2D class averages into a 3D map, all through this fabulous software like Relion, like CryoSpark, like System. So basically, we're doing that now at a rate of about 4,000 images in a 24-hour day. That's about 20 seconds an image, and that's because our cameras are limited and for some reason will not serve us images very, very rapidly. And because of that, this is our rate of data acquisition. This is where we got the first Krios, the second Krios, the third Krios, and recently sped the whole process up by doing some calibrations that allowed us to collect a little bit more rapidly every time we move the stage. So we're on quite a harsh trajectory, and it's going to get harsher or better. I'm very excited, because the new larger cameras and read out faster, they have larger formats. We will get at least 10,000 images a day. We hope 20,000 images a day. So going up by a factor of 4, more or less overnight. And these factors of 4 or 5 are quite hard to cope with, both on the processing side as well as the networking and the disk side. But this is a great problem to have. We're not complaining about this. It's a very good problem to have. But it also means all of that software has to suddenly get much more rapid if we're going to keep up and not cause bottlenecks. Okay, so in terms of those bottlenecks, of course, the real world is much more complicated than these toy problems I've been showing you, and I'll just show you a few case studies. One is quite old, and these two are brand new. And the first case study is to do with studying ribosome biogenesis, so a little bit different from Joachim, but at least everybody's familiar with a ribosome. And this is work we did for years, actually, with Jamie Williamson and some of his postdocs, and then some of our grad students. Actually, these were all grad students, worked on this project for a while. And what do we mean by that? Well, the ribosome is a fundamentally, profoundly interesting molecule. It's also a self-assembling machine. So the 70S ribosome is made up of a 30S and a 50S that come together. But this 50S and this 30S will self-assemble in the cell with a bit of help. So we need to understand how this amazing machine that assembles then every other machine in the cell, how does it come together? It's profoundly important to understand that. It's also important to some extent because of antibiotics, because if you can mess up the assembly in bacteria, you can stop them from multiplying. So it has drug importance, but also just fundamental importance. So the work was done many years ago in the 1970s, showing how these different proteins, so there's 20 or 30 proteins and a couple of big RNAs, and if you just mix them together, how do they come together? Who binds first? What binds to what? What exactly is the pathway along that? And these were all kinetic and energetic work that was done with sort of not directly visual methods. And Jamie's lab, together with us, wanted to do this structurally. We wanted to actually see this in action. So this is a complicated story, but the simple version is you kind of stall the cell a bit to start making kind of stalled intermediates, and then lyse those cells out, and you freeze the lysate just like this, and this is kind of what it looks like. There's all kinds of junk in here. There's these kinds of things which aren't ribosomes because it isn't a really clean prep. It's just more or less a lyse cell, but we can certainly pick out lots and lots of ribosomes from there. And then we just pick those ribosomes out, and then we go through that whole pipeline that I was talking about. You classify it out, and you turn those 2D classes into 3D structures. And in this case, by doing the sorting and classifying, it was found eventually that there were 14 maybe, at least 14 different conformational states of these intermediate ribosomes. So these ribosomes that were half-formed, and that's what we wanted to see, because that would tell us what that formation pathway was. But tons of problems in doing this, because how do we know there's 14? Maybe there's 1,400, maybe there's 14, maybe some of them could come together. Do we really know what pathway they're on? There was a ton of work done with mass spec and with all kinds of other biophysical techniques to try and say that this was the pathway, that the assembly pathway for these ribosomes can go down all kinds of different paths. It can dead end. It can cross over from path to path. But this had to be inferred using a lot of other biochemical information. It would be great to be able to do this manifold stuff and just say, okay, no, I can see this whole thing. I don't think this is appropriate for this work. But if there were more tools to tell us more about how we go from this step to that step. A second case study is this is done work with a grad student who just graduated from our lab, and Felipe Mancia from Columbia University, and they're interested in tuberculosis bacteria. Tuberculosis is a real scourge. In fact, it's the #1 cause of deaths worldwide at the moment. So it's very important to understand, how to attack the bacterium. And one way to attack it, the reason tuberculosis escapes our immune system is that the bacterial wall consists of a very complicated system. It's got 2 lipid bilayers, and it's got a gigantic, big, sugary sort of protective layer between those lipid bilayers. And one of the enzymes, one of the machines that builds that cell wall is this thing here called AFTD. And if you knock that, if you can interfere with AFTD, if you knock it out, the whole bacterium dies. So it's an essential molecule, and therefore it's a good target for drugging the bacterium. And then, we did a whole, Yongzi did a whole bunch of different structures, with structures bound to ligands and various things, and that allowed him to say something about the mechanism of action of this molecular machine. So that's very nice. What are the problems along the way? So this is a typical, probably a very good image, because it was put in the paper, of what that thing looks like frozen vitreous ice. Picking particles out of there is a challenge. The one I showed you, there's a particle, there's a particle. Here, what exactly is a particle? If you get really, really good at it, you can say that maybe and that, but then there's bits of aggregates and everything. So particle picking is still a bit of a challenge when you're doing these very difficult structures. Now that you've picked the particles, you turn those into these 2D classes. And now, how do you choose which are the good 2D classes? Right now, a lot of it is done manually. There's a human being staring at it, and they say, those all look good, but I don't like this one, I don't like that one, it's probably junk. How do we know it's junk, And how do we know these are the average of thousands of particles? How do we know that, 9,000 of them were good and the other 1,000 were bad, and that messed up your class average? So are we throwing a lot of data away at this stage and doing the sorting incorrectly? And we would love better methods to do that sorting. Once we pass on to the 3-dimensional reconstruction, then again we get multiple classes now. We don't ask for one structure, we ask for many structures. And then some of them are good and some of them are bad. How do we decide? Just by eye, usually. You just say that one doesn't look like any good, and these are the good ones, and then those good ones then get turned into this very beautiful high resolution structure. But it seems to me we've left a lot lying on the table there and there and there. Not only have we maybe thrown a lot of data away that we need, but we've missed a story too. Because many times, as all of the people doing cryo-EM will know, this is less than 10% of the particles that we started with end up there. What is the story in the other 90%? Is it some fabulous manifold story like Joachim's telling, or is it just garbage and junk that we have to discard? And we don't really know that yet. So that would be good to know. And then even at this stage, there's a whole bunch of super clever software that has to be used now, per particle CTF refinement, this non-uniform refinement you probably hear about from Ali, I think, at the end of the day, and beam tilt correction that Shaw's has incorporated into his Rely. And that's really what squeezes out these last few bits of resolution and lets us to go to sub 3. And sub 3 is an important milestone in cryo-EM because that's where you can really see a drug. So the pharma companies, they want to be sub 3. If it's 3.8 or 4, it's not so good because you can't really see the chemistry of how their drug is binding to this molecule. So these are important things that you have to do in order to squeeze that structure. And then lots of other interesting things in this structure. In the wild type, there was a thing attached to it. Doesn't really matter about the biology. In the mutant, that thing that's attached is very blurry because it's loosely attached. We would love to know more about what's going on there and be able to pull more information out of those kinds of images as well, rather than just look at the results. Okay, one final case study, very similar actually, and this is to do with malaria, which is the third worst disease in the world, kills very, very many people every year. And malaria is very important to understand this parasite because the current set of drugs that are being used are now, the parasite has become resistant. The original drug that everybody used was called chloroquine in the 1950s. It was incredibly effective. It was super cheap. And the parasite became resistant to that chloroquine. And it became resistant because of a mutation in this particular machine inside the parasite. And all that machine does is flip the drug. Actually, I've got a diagram. When you get this chloroquine drug in, this machine mutated to flip the drug out of the compartment where the drug was doing its work. So it's important to understand what that thing looks like because maybe we can design new drugs which would not be resistant. And we have exactly the same problem in here. This is a tiny, tiny protein. It's 45 kilodaltons. It's almost invisible. It's in a nanodisk. So the nanodisk actually weighs more than the protein. So if you solve this without a little fab attached, you've solved nanodisks really well to high resolution, but not so much a protein. So it's hard to see, hard to pick these particles again. They're not very visible. Again, how are we discarding the 2D class averages we don't like? We're making decisions. And then non-uniform refinement in particular, which I'm sure you'll hear about in the Cryoscope talk, was critical for this project. Without the non-uniform refinement, couldn't get that past blob level. And with non-uniform refinement, it went quite nice. And then with signal subtraction and all some of those other fancy things you'll hear about, it went all the way to 3.2 angstroms. And again, that's where you start to be able to think about druggability. Okay, so that's all the kinds of things we care about, very broad brushstrokes. I don't understand some of the algorithms, but I know what I want. So just a few things that we'd like, though. We want to improve automation, so we want to be able to pick particles much better. We want to be able to decide which are the good 2D class averages, which are the good 2D maps with some more help. I have thought neural networks might be able to help us with this for some time because people are doing this, but they're doing it a billion times. So we're capturing a lot of training data because people are doing it every day. And there's a program called Topaz. I think Tristan is here. I thought he was coming. Anyway, Alex will be here tomorrow. And these two, Tristan's a neural net person from MIT, and Alex is in our lab. And they've put together, for now, it's just a particle picker. What I love about this particle picker, most neural nets, you have to exhaustively pick all your particles and you have to exhaustively pick your not particles. This, the whole training data set, this is L delays. This is what our images look like. They're super, super, super crowded. And there's an L delays, and there's another 3 there, and that's how sparsely they picked this training data set. They picked very, very few, and then when they applied it, it picked everything. And it picked everything, and then, of course, they took this to a 3D map. Oh, here's the 2D class averages, and then to a 3D map, and it worked beautifully. The other thing it does is sort these things really well. So the sorting out tends to leave the junk particles at the end. So maybe we can do a more kind of clever thresholding of what we throw out just by choosing this. You can go on forever here. It can pick everything if you like, but you can threshold somewhere. And that's what I like about it, that it gives us a little bit more control. So where we see delays in our automated pipeline right now is we have to do a bit of fiddling around here, training or something at this step. We really want a generalized picker. After all these hundreds or maybe thousands of test samples out there right now where people have hand-picked and hand-curated, we should, I hope, be able to do this automatically. We should be able to automatically decide which ones we throw out, and we should be able to automatically decide how we proceed onto our 3D map. And if we could do all that, then as soon as we start collecting data, those 3D maps could start to be produced. And I'm not saying people shouldn't be involved. People should be involved at every one of these steps. But we need more automation, because once we get to collect 10,000, 20,000, 30,000 images a day, we're going to be solving 10 structures or 20 structures a day. So as much automation as we can get, we need, because you can't have somebody do this 10 or 20 times a day. The burnout rate becomes too high. So with an automated pipeline, all you're ever doing is moving the bottleneck from one spot to another. It used to be that the bottleneck was collecting the data. That's not going to be the case. The bottleneck is now for sure going to be analyzing that data in as efficient a way as possible, as you get as much out of it as you can. And that's the easy part. Much, much worse is doing all of this for cryo-ET, because now this is a really great tomogram. This is an easy one, because all it is some lipid membranes with these particles stitching between them that we care about. And those machines that are stitching between those membranes are the ones that tell your neurons what's self versus non-self. So when a neuron goes out to make a synapse, it needs to find another neuron, not itself, because if it just finds itself, you just have a short circuit. So these particular machines here are the ones that the neuronal system uses to detect self from self. So they're very important. And we wanted to look at them, and we wanted to understand where they were and how they were, et cetera, et cetera. This was already extremely difficult. We did use some neural net stuff from Steve Lutke's group, but it was very, very hard, and I think we need a ton of more improvement in order to make that even feasible and not be a huge bottleneck. And then, of course, when we go right inside the cell, like this example I showed you from our lab earlier on, it gets even worse, because now we're looking at an incredibly cluttered environment. These are all just little black dots. We don't know if that black dot is this thing or that thing. So we need a lot of help in figuring that out. Okay, just finishing up then. So for the single particle pipeline, I really think we're going to go onto the order of an hour from sample into the microscope to 3D map. That means we need a ton better software and a better automation, and it needs to be jolly fast. If you're doing 20 a day, you really don't want to spend 3 weeks processing each one, because you just make a bottleneck there. So you need to make this as easy as possible, and of course as easy for human beings to interact with as possible, because they have to be able to judge this. It can't be a complete black box, because we'll start making terrible mistakes. So you need to be able to be giving these interactions along the way and have a ton of validation as to knowing whether you got it right or not. And then for this pipeline, the tomography pipeline, where we end up with these very noisy tomograms, it's really hard making the sample. That's, again, probably not the topic for this group. But collecting the tilt series, not so bad. We can do that fairly fast. But of course, as soon as we do it fast, we need to analyze them really fast because there's just another bottleneck. And then this process of interpretation is ghastly. It takes months and months, sometimes years, for people to analyze these things. And that's where I really hope some of these neural nets or deep learning may be able to start having some impact, with all the manual training that's been done. And in this area, we're making some progress, but it's pretty slow. Okay, so this is the group at the Simons Electron Microscopy Center. It's a really super fun place. And we thank the Simons Foundation for our funding. We have a group of technical staff that keep our instruments running all the time. By the beginning of next year, we will have 7 Cryoses at the center, so it's a big operation. We have people who develop the software, these automated processing, etc. And then we have an amazing group of postdocs and grad students who are the engine that drive it, because they're trying to solve these biological problems, and they're the ones who need the new technology in order to be able to do it, and to be able to do it as efficiently and effectively as possible. Thank you for your attention.